Hello and welcome back to this Virology course. This is the second lesson. I'm talking to you about the genetics of the viruses, in particular the replication mechanisms, the interactions in between viruses and the ways of counting viruses in biological samples. The specific noun we use when we are talking about counting viruses in a sample is titration. So first of all, let's talk about the replication mechanisms. Well, as I told you last time, there are two kinds of viruses, DNA viruses and RNA viruses. So DNA viruses normally send their DNA into the nucleus and use the DNA-dependent RNA polymerase of the cell. But there are some kinds of DNA viruses which have a virus-specific DNA-dependent RNA polymerase that produces messenger RNAs from the DNA. Then, messenger RNAs are sent to the cytosol where they are recognized by ribosomes and translated into proteins. And what about RNA viruses? Well, the viruses which have a positive single strand, they don't need to do anything because their RNA has all the characteristics of a messenger RNA. So it is immediately recognized by ribosomes in the cytosol and translated into proteins. Differently, the viruses which have a single-strand negative RNA need to convert the RNA into a positive strand because the negative polarity of the RNA prevents the RNA from being recognized by the ribosomes. Now let's talk about the interaction in between viruses. First of all, I have to tell you that mutation in viruses is a certainty. It's not a probability. So, mutation in viruses is really important because it helps them to survive, to adapt to the environment, and to resist from the attack of the host organisms. Mutations can occur because of mistakes in replication or because of interactions in between viruses. Supposing we have two different kinds of viruses in the same cells. The interactions we may have are gathered in two groups. The first group is genetic interactions. The second group, non-genetic interactions. In the first group, we have basically recombination and genetic reassortment. In the second group, we have intrinsic interference, induction of defenses of the host organism. Then we have phenotype mixing, and finally, we have intragenetic and intergenetic complementation. So, what is recombination about? It's about an exchange of genetic fragments. When we are talking about DNA viruses, this exchange occurred by a sort of cut and paste mechanism. When we are talking about RNA viruses, the mechanism is different because it's up to the RNA polymerase that first uses a certain template to transcript the messenger RNA and then uses another template. So the messenger RNA we have is a mix in between the first virus and the second virus, because the RNA polymerase used two different kinds of templates, one coming from the first virus and the second coming from the second virus. So we'll have mixed proteins and finally a mixed virus progeny. And what is genetic reassortment about? This mechanism occurs only in RNA viruses which have a fragmented RNA, and it consists on an exchange of genetic fragments in between two RNA viruses. Now let's talk about non-genetic interactions. As I told you, we have intrinsic interference. This is a simple mechanism. It consists on preventing an infection from another virus. So think about it. This is a functional mechanism because the presence of two viruses in the same cell will be dysfunctional because both of the viruses will have less energy, less molecules, and less probability to replicate normally. So the first virus tries to prevent a superinfection by using all the energy it has, or by occupying all the ribosomes, or finally by inducing the cell not to synthesize the receptors recognized by the second virus. Another non-genetic interaction is the phenotype mixing. This mechanism consists on synthesizing the pericapsid of another virus. As I told you, the most important part of the virus is the pericapsid because it has a lot of glycoproteins, a lot of proteins essential for the infection, but also for the recognition of the virus by the host organism. So, what happens if a virus has got a pericapsid belonging to another virus? It happens that the virus is able to infect a wider range of cells, and it is recognized to be the other virus and not itself, because the host organism doesn't know what's inside the pericapsid. It doesn't know that inside the pericapsid there is the genome of another virus. The host organism thinks to be infected by the second virus, not by the first one. So, when the virus the first one infects a cell, it's free to replicate and to do whatever it wants because the host organism has synthesized all the antibodies directed to the second virus. Another non-genetic interaction is the induction of the defensive of the host organism against the other virus. The last non-genetic interaction is the complementation. This mechanism consists on the fact that two viruses inside the same cell have more probability to replicate instead of being alone. What I mean is that the viruses alone wouldn't be able to replicate without the presence of the other one. I want to end this lesson by talking about titration. So, how to count viruses in a biological sample? We have two main ways. The first one is the serological titration. The second one, the biological titration. The serological titration consists on counting viruses by considering a specific mechanism. The most part of viruses have got glycoproteins on the surface which are able to hemagglutinate red cells. So by counting the number of hemagglutinated red cells, we have the number of viruses, because hemagglutination is directly proportional to the number of viruses. The second mechanism consists on counting the lysis plates we have in a cell culture. So, in laboratories, scientists normally put together in the same cell culture cells and viruses. Then they normally wait for an hour and they observe the culture. What do they see? They see empty spaces in the culture. The empty spaces are the mirror of the cell's death caused by viruses. Normally, viruses are not able to move inside the cell culture because of the presence of agar. So, by counting the number of lysis plates, we have the number of viruses. They also are directly proportional. So, I hope that you're enjoying this fabulous, mysterious, microscopical world. See you in the next lesson. Bye!